Hello future doctors, welcome back to my channel Quick Neat Botany. In this session, we are going to discuss some key points of molecular basis of inheritance chapter. In this chapter, we are going to understand the details of genetic material, which is responsible for the transmission of characters from one generation to other generation. From one generation to other generation. Let us see the historical events of this genetic material, contribution of the scientists in its discovery. First, in the year 1869, the scientist Frederick Mischer he observed a substance. He identified first, he observed a substance in the nucleus and he named it as nuclein. So we can say the first name of this genetic material. This is the first name. Later on, the studies of Altman. Altman, he observed the acidic nature of DNA or acidic nature of this nuclein. Due to the presence of phosphoric acid, it is acidic in nature. So, he named it as nucleic acid. The term nuclein was given by Frederick Mischer. The term nucleic acid was given by Altman. There are two types of nucleic acids in the living system. These are the DNA, which is deoxyribonucleic acid, and the second one is the RNA, ribonucleic acid. Let's see the significance of this DNA. It is the genetic material. DNA, it acts as genetic material in living organisms. In most of the living organisms is given, but we can say in all living organisms. Not only most of the living organisms, in all living organisms. No organism with RNA as the genetic material. Some viruses are having RNA as the genetic material. Of course, viruses are not living organisms. They are just living particles. And RNA, it acts as genetic material in some viruses. It acts as genetic material in some viruses, but not in living organisms. It is present in living organisms, but it does not act as genetic material in living organisms. Let's start our discussion with the DNA. This DNA is a polynucleotide chain. It is the largest biomolecule, and this is polynucleotide chain. That means it is made up of many subunits called nucleotides. These are the subunits of structure of this DNA. Let's see what is that nucleotide. It has three components. That nucleotide is having three components. These are, the first one is phosphate group, or we can say phosphoric acid. The second one is pentose sugar. The two here, the sugar name is deoxyribose sugar, which is a 5-carbon sugar. It has 5 carbons. And the third one is nitrogen containing base. Let's see the structure of this nucleotide in detail. Here I am taking phosphoric acid structure. This is phosphoric acid. Here I am taking deoxyribose sugar structure. It is a 5-carbon sugar. First carbon, second, third, fourth, and this is the 5th carbon. Here it has H, OH. Here H, H. Here H, OH, H, OH, H2. Its formula is C5H10O4. And nitrogen bases are of two types. Nitrogen containing bases, purines and pyrimidines. Let's see about purines. Purines are heterocyclic compounds and they have two rings. So we can say dicyclic compounds, heterocyclic and dicyclic. They have two CN rings in the structure. They have two rings which are made up of two different elements. That's why heterocyclic and dicyclic. Both the elements are different, carbon and nitrogen. If you see that structure, out of two rings, one is hexose ring and other one is pentose ring. Hexose ring and pentose ring. See, this is the first position, second, third, fourth. First position, second, third, fourth, fifth, sixth, seventh, eighth, ninth. In its first position, third position, seventh position and ninth position, they have nitrogen. And in remaining positions, it has carbon. Here two types of purines we can see, adenine and guanine. These are purines. Let's see pyrimidines. Pyrimidines also heterocyclic. These are also heterocyclic, but these are having only one CN ring, so we can say monocyclic. That means they have only one CN ring in the structure. That too it is a hexose ring only, no pentose ring in the structure. That is hexose ring only. So numbering from here 1, 2, 3, 4, 5, 6. In its first position and in its third position it has nitrogen. In remaining positions it has a carbon. So carbon, nitrogen, two elements are there. So heterocyclic. Dicyclic and heterocyclic compounds, purines. Monocyclic and heterocyclic compounds are pyrimidines. Here in DNA, two types of pyrimidines, thymine and cytosine. Thiamine is vitamin, but here it is thymine, which is a nitrogen base. One more pyrimidine is there, that is uracil, but it is not there in DNA. It is present in RNA. Let's see how these components in the nucleotide are connected with each other. If you see this phosphoric acid and sugar Both are connected with each other by ester bond, or we can say phosphoester bond. There is phosphoester bond between phosphate and the sugar. Nitrogen base attaches to the sugar by N-glycosidic linkage or N-glycosidic bond. The bond between sugar and the nitrogen base is N-glycosidic bond. The bond between the phosphoric acid or phosphate and the sugar is phosphoester bond, or simply we can say ester bond.